Good morning. My name is Lauren Hemsworth and I would like to start by acknowledging the traditional owners of the land on which we meet, the Wurundjeri Woiwurrung and Bunurong peoples. I would also like to pay my respects to their elders past, present and emerging. I would like to welcome you to the first Animal Welfare Science Centre for 2024. It is the first time that we are meeting with the wider animal welfare science community since the passing of our director, Professor Andrew Fisher. And I would like to take a moment just to acknowledge the significance of his loss, both for the centre and for the wider scientific community. We will, in a coming seminar, be paying tribute to Andrew's contribution to both the Animal Welfare Science Centre as well as the wider animal welfare science community. Andrew was a big supporter of the Animal Welfare Science Centre seminars, so I'm pleased to announce the restart of seminars in 2024. And I'm looking forward to the tradition of being able to facilitate interesting and engaging discussions around key animal welfare issues, to promoting our early career academics, and to facilitating rigorous scientific discussions. So I'd like to now start by handing over to the Centre's resident chicken expert, Dr. Peter Taylor.
Thanks, Lauren. Of course, it takes quite a few people to organise a seminar, not only those within the centre, but also the university. So start by acknowledging Melbourne University's support in this space, and also U4. So U4, through their LINK program, have partially funded some travel costs and to get some fantastic speakers with us today. So I'd just like to acknowledge U4 for their contribution to today's seminar. Today's seminar will be recorded and that includes any discussions that we have. We do ask for discussion and active participation from you. We want to hear, we don't want to just share the science today, but we also want to hear from you, for those people who work with and advocate for poultry welfare. So we want to know your perspectives and considerations, and that in that sense we're going to have some time. At the end of each of our talks we'll have two presentations today for questions. So we have Mia Cobb who will be monitoring the chat today, so feel free to put your questions through the chat, and of course you can put your hand up and we can have those online. We also want to have some anonymous input from you, and we thought we'd do this via a poll. So just to get us started, get us to know each other a little bit more, please scan the QR code, or you can text in an answer, or of course you can go to pollev.com/AWSC seminar. And tell me what you think about my chicken dress. So for those online, I'm not sure if you can see it, it is a great arrangement of jungle fowl, roosters, hens. We want to know, is it incredible you need more poultry clothing in your day-to-day life? Would you prefer, would a dog canine dress be better? It's not bad. I've seen better. You're in a safe space, so please contribute honestly to these polls. Remember, they are anonymous, and of course you can opt out in any of the questions we ask today. So if you prefer not to say, feel free to, you can see the chat, an A definitely. Now, let's have a look if we can see some of the responses. All right, we've got 89% saying absolutely poultry clothing is a must for day-to-day life. We've got the right audience here. 6% dogs would be better.
Wasn't me.
I was thinking Mia Cobb. No. Our poultry welfare guest, Dr. Mike Toscano. Not the answer I was expecting. Not bad. We've got a few who are perhaps having some issues with the technology.
It's too far.
It's too far away.
QR codes, you can't read it.
Oh, it's too small.
Comment sections are like A's.
Lots of A's. Okay. If the QR code is too small for those in the room, you can join by web, pollev.com/AWSC seminar. But we're going to move on. I want to hear a little bit about you. Where are you joining us from today? I know a bit about chooks, but my geography is not great, so I might need some help with some of these tags. Tasmania, great. New South Wales. Oh, here we have our first overseas international participant. Can someone help me out? Where is that? Oh, we've got some in New Zealand. Hello, New Zealand. France? Europe?
It's a bit hard to drop a pin on the phone, so I think I put myself in the ocean.
We have someone in the ocean using the last of their battery to join the seminar. Good choice. Well, wherever you are today, whatever time zone you are in, we welcome you. What we'd like to hear now is one thing that you'd like to learn more about today. We're here, we're discussing poultry welfare. We're not talking about meat chickens today, so that could be something you would have liked to have learned. We are focusing on hen welfare. We have one participant who is really interested in piling. It's lucky we have two experts in piling and smothering. Environmental enrichment, responsibility of the retailer. Poultry welfare, fantastic. If you like other responses, you can give them a thumbs up. What are the current standards for layers?
Botulism in the comments.
Botulism. Anyone covering botulism today? Okay. Enrichment, we've got 4 thumbs up for enrichment. The current standards are for layers. We are going to hear a national perspective and also an international perspective as well. What are some of the welfare-specific considerations here in Australia, but also what's shared globally and where is the science at? Great. I thank you for your contribution. We are going to come back to this poll at the end of the seminar and we will take on board your comments. So thank you. And just one last question before I introduce our first speaker. How important is poultry welfare? Is it extremely important? Is it important? Somewhat important or not at all important? Time is precious. We have 90 minutes together today. We're going to pack in a lot. So I imagine if the answer was D, you wouldn't be here. But you're in a safe space. You can share. So we've got extremely important, 79% of our participants, 21% said important. Excellent. I'm going to ask you some specific questions after the seminars, but please keep this interaction. I know that we have many participants joining us online. We have a few here in this beautiful space at Melbourne University. So please feel free to put some comments in the chat, and at the end of each of the presentations we'll make some time. So it is my pleasure to introduce Maxine Rice. Maxine Rice is our first presenter today. She's a PhD student within the Animal Welfare Science Centre at the University of Melbourne, and her PhD is investigating piling behaviour in Australian free-range laying hens. Before embarking on a PhD, Maxine completed a Master of Philosophy investigating shy feeding behaviour in lambs in feedlots. Maxine has also worked as a research assistant within the Animal Welfare Science Centre for 17 plus years, and her work has largely been focused on observing behaviour in relation to animal welfare, and not just in poultry, across a range of species, agricultural, companion and zoo animals. So thanks for being here, Max, and we look forward to hearing from you.
Thanks for the introduction, Peter. Okay, so as you can see, my topic for today is smothering. For those of us in the audience who might be naive to what smothering is, this is the name that we give for mortality that occurs as a result of piling behaviour. So what's piling behaviour? Well, piling behaviour is when birds press up together. This often happens in laying hens, mostly in free range, where they press up together in such a way that some of the birds can become asphyxiated and subsequently die. This is a problem that is occurring not just in Australia, it occurs worldwide as well. It's something that has really in the last decade become of great interest to producers and researchers alike. Most of the research around, particularly around smothering, has occurred in the last 10 years. And I just thought I'd give you an overview here to sort of show roughly what type of, or what quantity of mortality are we talking about here. And as you can see, it can range anywhere from only, is this pointer working? Yeah, you can see my, yep. From 1.6% of mortality right up to 26.66%. The most recent study on smothering I'll cover in this presentation here was conducted in Australia over 84 flocks, and it's quite a substantial piece of work. So it's important to note that there are 3 recognised types of smothering. The first one is nest box smothering, and it's quite a specific circumstance where you have a number of birds crushing into the same small space of a nest box. Generally when birds have a preference for nest boxes, they can all try to use it at the same time, and that can result in some mortality. This generally results in 1 or 2 birds per event dead. The next type of smothering is panic smothering. So this largely results as from a disturbance in the shed, something startling. It could be a predator, or it could be a loud noise, a sudden appearance of a light. This type of smothering can result in quite large numbers of bird deaths per event, and can be considered quite a problem, but generally producers felt that they know the cause of what the panic was, and so that's something that can be addressed. So one of the more concerning types of smothering that producers have is what we call creeping or recurring smothering, and this type of smothering can sort of each pile will develop slowly and progress into quite a substantial pile at times, and it generally, once it's started in a flock, it'll reoccur through a flock throughout production. This can result in quite large mortality or quite small mortality as well. So anything from 10 to 50 birds or more per event is fairly common. So it's potentially not as large a number per event as the panic smothers, but it is also quite a considerable issue. And this is an example of quite a substantial pile that we've observed in an Australian flock. And this pile lasted for over 3 hours. So it can go on for quite a long time. This pile itself did not actually result in a smother. So that's another important thing to note, is that piling behaviour does occur more frequently than smothering, and so that's one of the reasons why a lot of the focus that has been done around this topic has focused predominantly on piling, because that is something that you can observe, whereas smothering is something that most people will find the resulting dead birds and have very little extra information around what's happened there. So looking at piling behaviour in an Australian flock, we ran a small study that looked at one flock. We had one camera over the scratch area. This is the area where the birds can go down in duff spay that has a littered area. And we tried to identify any triggers or causes that we could see of piling based on videos alone. So this is not to say this is what actually caused the pile, but these are events or things that we noticed around the same time as the start of the pile. And what you can see here is that predominantly it's really difficult to identify what causes a pile, just visually. So 48% of the piles that we observed, we couldn't detect anything that could have potentially started that pile. However, 41% of the piles occurred when there were shards of light coming in through the side of the shed. So that was where sunlight was sort of beaming in through the windows, and the birds were quite attracted to the light. 8% were caused by a stock person moving through the shed. So that the interaction with the stock person could also be a cause for concern. And only 3% were what we would have identified as a panic smother caused by a sudden bird fright. So this is just to give you an idea of what it looks like when the sun's coming through in the shed. As you can see, it's quite bright in patches, and there's quite considerable interest from the birds. This generally happened in the afternoon in this flock, so the afternoon sun would come through the shed curtained sides. And so that might be one of the reasons why we might see more piling occurring in the afternoon, and that has been confirmed by studies coming out of Michael Toscano's group as well, in terms of when piling behaviour generally occurs. In terms of the stock person, you can see that in this image here, there's very few birds around the camera here, but there is a stock person starting to move through from the top of the field of view. And as he moves through, you can see that the birds continue to follow him until he's gone through the gate, and they press up against the gate there as well. So the birds, and you see this anecdotally when you're in the sheds, the birds are really quite attracted to people, so you can't really stand still for too long without causing some piling behaviour. So as I said, predominantly the piles were occurring in the afternoon. And interestingly, the piles that occurred in the afternoon were significantly longer in duration. And that could be because many of the piles in the afternoon were associated with that sunlight coming through, so that the trigger that's there is quite a persistent trigger. But that's something that we still need to investigate. Only 2 of these piles, of the 75 piles we saw, resulted in smothering. And of those 2 that we saw, the 2 smothering events, neither of them were detected by the stock person, so they weren't reported in their daily smothering reports. So the problem with smothering is likely to be underestimated. So that brings us to a large longitudinal study that we conducted, where we looked at 3 separate farming operations. 2 of them were in Queensland, 1 was in Victoria, so they're quite spatially different, with different climatic conditions. We had 2 different housing systems represented, so the Avery system, which is a multi-tiered system, and a flat deck system where all of the birds' resources are provided on one level. So an example of the Avery system is on the left here. So you can see there's different levels in this structure with all their feed and water provided on those different levels. And then the flat deck system, which is this one here, which is all on the one level. Some of these also had winter gardens as well, which is an outdoor area that's under cover, but otherwise exposed to the elements. We did have different breeds represented. Unfortunately, though, we predominantly had highline browns, so we weren't able to say too much about the breed effects within this study. And our flock ranges from 16,000 up to over 40,000 hens per flock, so they're quite a range there. The data that we collected was done by surveys with the flock managers. We also drilled down into their farm records and had some weather stations on each site, and we conducted behaviour tests looking at the responses to humans and also to novelty. So these pictures here just give you an idea of the variation in smothering that we find in the different locations of smothering within each shed. So this is just a selection of some of the sheds. I think it's 2 from each organisation. And as you can see, some sheds have it occurring pretty much widespread on both sides of the shed and all through the middle of the shed, whereas some sheds are pretty much on one side of the shed. That could be something to do with the orientation or something happening around the range of the shed, since this is mostly on the outside. But just to give you an idea that it's really hard to predict exactly where the smothering is going to occur based on this information. What we did see was that because we asked the stock people to note down whether the smothering was occurring inside or outside the shed, we found that of the 46,639 smothering events we recorded, 12,751 were inside the shed and 19,282 were outside the shed. We did have a considerable number that were where they didn't report the location, but it does appear that smothering is occurring almost equally inside and outside of the shed. We didn't see any effects of or any patterns in relation to the season in which smothering was occurring. So these graphs show a frequency histogram showing the number of deaths by calendar date, so the calendar date across the bottom, and the different bands here represent orange for spring and summer and blue for autumn, winter. So it's really difficult to see any kind of pattern occurring there. So we looked at some of these variables that we've collected in terms of their relative risks in relation to smothering both indoors and outdoors. And what we've found is that with the increased water to feed ratio, there was a decreased risk. And so the way this graph works is that if we hold 1 as a standardised risk, then anything below 1 is a decreased risk, anything above 1 is an increase in risk. With outdoor is the orange points and indoor are the blue points. There was also a increase in risk of outdoor smothering when there were fewer shed walks being performed. And quite interestingly, in terms of the shed type, there was almost 2 times or more than 2 times the risk of smothering for both indoor and outdoor smothers in an aviary shed when compared to a flat deck shed. Now, in this case, the aviary shed, it encompasses quite a large number of differences between the flat deck and the aviary. So you've got that it's a multi-tiered structure, but there's also a lot of other differences that are related to that. So there's differences in terms of the management of these sheds, the number of times that people go in and out, the different ventilation types that they have for these sheds as well. So we don't really know exactly what it is about the aviary shed that might be causing a higher risk, and that's something that certainly needs further research. And in fact, all of these relationships here, we don't consider them to be causal relationships. We don't know if they're causal, so we need to have more research to look into what exactly is causing this relationship. In relation to the climatic factors and environmental factors, more smothering was occurring during rainy days and also in days of high humidity. So that's greater than 70% humidity, and any rainfall at all had an increased risk of smothering. Interestingly, when the temperature was greater than 27 degrees, there was a higher risk of smothering inside the shed compared to outside the shed. And this could be an indication that there's more birds inside the shed on a hot day. I'd rather be inside if it was a hot day as well, so that could be just a bird density relationship. But again, that's something that we need to drill down on a bit more closely. And in relation to the bird's behaviour, we found that birds with a higher fear, or flocks with a higher fear of novelty, had an increased risk of smothering, and birds with a lower fear of humans had an increased risk of smothering as well. And so that might speak to the reaction of the birds to the stock people and the attraction of the birds to the stock people, leading to a higher risk. And I'll just cover this briefly because I've only got 1 minute. We did briefly have a look at smothering during rearing, and this was just with the limited data set collected via surveys. And we found that 44% of all the flocks that we looked at had some form of, or some amount of smothering during rearing. It was only a small amount, so about up to 2% of the birds that were placed. But it was occurring between 3 and 15 weeks of age, so it's really quite early on, and something that we should be looking into further. And so the key take-home messages are that within Australian flocks, we found that 16%, or smothering accounted for 16% of the mortality in our flocks. It's occurring in rearing as well as in production, which is something that we weren't aware of when we started this project. And the identified risk factors included the shed type, with aviary being higher risk than flat deck, fewer shed walks, less fear of humans and more fear of novelty, extreme high temperatures and moderate temperatures, and high humidity and rainfall are all considered to be risk factors. But again, we need more research here to identify if these are actually causal relationships. And piling occurs more frequently than smothering. It occurs more frequently in the afternoons, and the piles in the afternoons have longer durations. And one of the things that we do need to look at is what is it about piling that might put that pile at a risk for smothering. So how do we judge whether a pile is a good pile or a bad pile? Yes. That's thanks to the research team and all of our participating organisations and Australian Extra Funding.
Amazing, Max, thank you very much. It is really interesting to look at. I think what we're going to hear today is different methods and the pros and cons and what we can take and interpret from that data. And one of the challenges of conducting some of these things on commercial farms. It's relevant, but talking about causal factors where we need to go from there.
Time for one quick question.
One quick question?
One quick question that's come through from Hannah. Has there been any research on the minimum flock size where smothering or piling begins to occur?
No, not to my knowledge. There has been, there was some relationship shown in, I think it was British flocks, that smaller flock sizes were potentially, less smothering occurred in the smaller flock size. But it wasn't specifically an experimental setting.
Well, that was a nice quick question, so thank you, Hannah, online. And I'd just really like to thank Max for sharing all of that data. And I was going to say something about you piling around a computer to watch those behavioural observations, but I thought it was in poor taste, so I won't. Thanks, Max. We will have some additional time at the end of Mike's talk, maybe for some additional questions. It is my absolute pleasure to introduce Dr. Michael Toscano. Mike is the group leader at the Centre for Proper Housing of Poultry and Rabbits at the University of Bern in Switzerland. He was born and raised in the United States, where he completed a PhD at Purdue University, and was held his position in Switzerland since 2013 after 7 years of postdoctoral research at the University of Bristol in the United Kingdom. He's attracted over $30 million in funding from multiple funders in Switzerland, the US and EU, and his research focuses on all aspects of welfare and housing for poultry and rabbits, where his methodological expertise include behaviour, physiology and management, but not PowerPoint. Leadership in these areas, I will...
Oh, I'll just let you do it.
Let me pop this up. We're going to share your...
You want a hand?
Here we go. Leadership in these areas have been prominent, including roles in the EU cost actions and focusing on livestock phenomics as a working group leader, and keelbone damage net, coordinating keelbone fracture research and training activities as chair. His specific research includes the use of sensor technology towards better understanding animal behaviour at the individual as well as the group levels, and how that information can be used to improve breeding of laying hens for cage-free housing systems. Additional noteworthy topics that he's focused on is determining appropriate feeder space, improving depopulation at the end of lay, and identifying causal factors of piling behaviour and also toe pecking. His research contribution in the area of animal welfare has been recognised by the Poultry Science Association, which awarded him their poultry welfare research in 2016. He's a long-standing and active collaborations with multiple industry partners, including the world's largest manufacturer, cage-free housing and suppliers of laying hen genetics. It is my absolute pleasure, and with much gratitude for making the big flight to come out here and be with us, that I welcome Dr. Michael Toscano.
Thank you, Peta. I couldn't have written that better myself. First, I just wanted to say thanks to Peta. I've known her for a while now, and it's been a really great collaboration. I've always really enjoyed working with her. I like her style, but more recently with our parallel work in piling behavior, we actually were able to form an actual collaboration, and that led to Maxine actually coming to Switzerland, and Maxine was also did incredible work with our group. And then now with the Australian Eggs Grant, it looks like we have a great future ahead of us. So I want to say thanks to Peta. I also want to say thanks to UFA as well as PICS for funding to bring me out here as well. And if you like what you hear today, I'm actually going to be at the PICS conference. Hopefully I'm saying that right, PICS. I'll be at the PICS conference next week. So if anybody would like to chat with me, I don't really know much people in Australia. So I'll be sure if you would like to chat, please let me know. So thanks to all those people. And for today's talk, I really first wanted to talk about an approach that actually I apply for my group, the ZTHC, but also I imagine is familiar with other people. But I wanted to talk about what we see as the fundamentals for research, and then how we can really take this information and build housing for the birds. And then I'm going to be talking about this concept of individualized behavior, and really what it can tell us, and what it has told us more recently. And then lastly, I wanted to talk about a rather large project what I call HenTrack, that's really fusing welfare and technology to use that towards breeding. And I specifically wanted to talk about this idea of piling that involves this collaboration that I have with Peta. So just as an outline for what I'm going to be talking about today. So just as a research philosophy, and I guess this is something that many others have as well, but I think it's important to cover it today because it's also going to frame, I guess, the approach that we take and the particular reasons for why we do this type of research and why we built this experimental design and what drives really the research. So the idea is that we should understand, we should conduct the fundamental research that should then inform the applied interventions. When we are looking to solve, say, the problem of toe pecking, we need to know why the birds are toe pecking, what's motivating them. Otherwise, often our interventions can be quite spurious. So our projects are really developed to understand these mechanisms behind these types of problems. And then we can test these solutions and ideally apply them on farm so we can deliver actually true changes. We also try to use relevant housing. And I think this is a big difference, particularly for poultry. What you observe, say, in small groups, in 5 groups of birds, may not or is unlikely to apply when you then try to work with 30,000 birds in a house. So I had a bigger slide that I cut out on this, but for most of the research that I'm showing about today, understand that we're using groups of birds that are anywhere from 200 to 360 birds, which is still pretty small, but I think it's very different than until you're 5 birds in a pen, as well as commercial houses, which we also work with as well. So an idea that for these projects, when we're conducting them, about trying to understand the fundamental mechanisms, we can then apply, our interventions are ideally going to be more helpful. So, for example, we've been working with the ramps to improve locomotion and reduce bone fractures. What is motivating the birds to pile, as well as why are they piling, or that they are piling. And also questions like this problem of toe pecking, which I'll cover, where are the birds actually pecking themselves? Are other birds pecking them? Is it a combination of it? So I think for each of these problems that we see, we really need to understand what's driving it behind it, so that we can then say, okay, if the bird is self-toe pecking, then it must be uncomfortable, or maybe it has dry skin, which is very different from birds that, say, are attacking other birds. So it's really important to understand those mechanisms. And luckily, with the facilities that I have at Switzerland, we can kind of do both. We can work with small groups, we can work with large groups, and then extend that all the way out to commercial settings. So, as I suggested, I think it's really important to understand the bird's needs, and because my background is behavior, from a behavioral perspective. I've been lucky enough to have lots of collaborations with people that do genetics, epigenetics, microbiology, which I think is really important to integrate. But my particular focus is on understanding the bird's needs from a behavioral perspective. And this has really led to this idea that we should really be building the house around the bird. I remember back from my master's, working with Ted Friend at Texas A&M, and he said, we had these gestation crates that are square because that's what architects like. But that may not be best for the animal. And I really think we can actually see this a lot. Rather than build nest boxes of a certain size, because that fits the dimensions of what the architects like and the cost of the sheet metal or whatnot, really what does the bird want? And while the costs are really important, we really need to understand what does the bird want, and that should be guiding us. And this is actually important for Swiss legislation. I won't translate it, but the idea is that housing should not prevent the normal biological functioning of the animal. So there is nowhere in Swiss legislation that says that cages are banned. But to provide housing for hens, because we assume that the birds want to dust bathe, because they want a covered area for a nest box, you need to provide housing that provides that. And it's simply not possible with cages to provide, say, litter. But in theory, you could actually have a cage if you can provide these basic things. So it's really important to understand what the animal needs, whether that's provision of litter, covered nest boxes, or etc. So one example I wanted to talk about is ramps, because this is a topic that we've been working on for a while and we think fits really well. It's always bad to pick an example that fits your your data very well, but I think it's a good one nonetheless. That's left over. So ramps, this originally started with a lot of work that I was doing with keel bone fractures. For those that don't know it, this is a radiograph of the bird's keel, and here's a keel here. It's an extension of the sternum. And when you look, say, here, you can see these massive breaks in the bone. I say here, there's lots of little ones here, and this big one there. So anywhere from 30 to 100% of the birds will have fractures, say, by 50 weeks of age. So this is definitely a big problem and something that I've been working on for a while. And before I got to Switzerland, I had a colleague, Ari Stratman, who was actually doing a part of this work as her thesis. But, oh, sorry, I'm getting ahead of myself. So first is just for people that maybe don't know poultry or don't know fractures, what's causing it? Because obviously we don't want birds to have this damage. So there's various explanations, and I put up a link for a recent paper that we released on explanations, but there's various ideas about egg laying, that actually as the egg is laid, it makes contact with the keel and causes damage. There's also this idea that birds are coming into lays too soon, and their bones aren't given enough time to develop. But the one that we're focusing on is really collisions. And I would say this was the main factor that we believed was causing fractures. A lot of it came from work out of Bristol with Lindsay Wilkins, where this is the height of all the things in the barn on the y-axis, and on the x-axis, this is birds with broken, the percentage of birds in that flock that had broken keels. And you can see that as the height increases, you tend to see an increase in the percentage of birds with fractures. So the idea is that in these open systems, the birds have access to perches, nest boxes, other things, and as they're flying, they're crashing into those things and causing these damages. So that is what we believe is a major factor. So our idea was, okay, if the birds are moving around and knowing what we know about behaviors, the birds are not good flyers. Okay? If a bird is, say, this is a typical aviary, if the bird is up here and it wants to go down to the litter, well, how is it going to do that? It could fly down and then it smashes into this wall, or maybe it tries to somehow navigate and then it comes back and then flashes into this perch here. This is what we think is causing the damage. So, but my colleague, Ari Stratman, back in her PhD, and again, this is before I came to Switzerland, so don't want to take credit where it's not deserved. As far as her PhD, she installed ramps in the aviary, which you can see here. So now if the bird wants to fly down, or wants to move down, instead of flying, it can walk. And looking at the percentage of birds with fractures on the y-axis and the age, and you can see for the control versus the ramp group, the birds with ramps had less damage. So this idea is like, okay, the birds have less damage, that's a good thing for the bird. And actually, when you think about the bird's physiology and its biomechanics, we actually worked with a really interesting person out of Montana, Brett Tobalski. He's worked a lot with Alexander Harlander as well. And when you look at the biomechanics of the bird and its ability to generate force, the bird is not very good for navigating. When birds fly, they get scared and they go up, and then they gradually come down and glide down into a big field somewhere. They're not like, say, a hummingbird or a sparrow, which can gradually move, okay? These are big, chunky birds, and they're not easily able to move from here to there. So they're not well suited to move in a tight environment like an aviary. So this works good for architecture, maybe doesn't work good for the bird. But these ramps, okay, they can move there. But what do the birds think of them? So this is a project. It's now actually, it's been submitted, and I have the reviews from the reviewers. Maybe it's one of you sitting on my desk, which I have to respond to. So, but this is a project we did in our commercial barn, where it's a commercial aviary, and you're going to see this picture a lot today. But what we actually can see, this is all the downward transitions with the ramps at our specific viewing points. And we had several groups, and so let me explain this. This group here is groups that had ramps in lay only. And then this group here is that groups that had ramps during rearing as well as lay. And then this is observations that occurred with lights on, midday, and then at dusk. And what this is basically where the birds started the transition. So highest would be birds that came from here and then moved down. Middle tier would be birds that started here and worked their way down, and then lowest would be from here and then worked their way down. So, and actually let me set this up because you're going to see this photo a lot. This is a typical aviary, and we've divided into these 5 areas. So the 5 would be the highest tier, and this contains feed, water, and perches. Zone 4 is the middle tier. Zone 3 is the lowest tier with feed and water. Zone 2 is the floor with litter, and then Zone 1 is a winter garden that contains water, perches, as well as litter. So this is a typical aviary, and we think about these specific resources in each of these barns, or each of these areas. So what struck me out here, and what we really liked, independent of anything to do with fractures, is the birds are making the transitions with the ramps. So this is just a percentage of downward transitions with the ramps. And what you can see here is even though the birds can easily jump down, they have the musculature to do it, still more than 70% across all time points are made with the ramps. So 70% of the time the birds are using the ramps even though they can fly down. And I thought what was particularly interesting is actually even from the lowest tier. So the lowest tier is right here. This is maybe a meter. So the bird could easily, say, jump down, but they're still using the ramps. And then this is now in lay, but we also see the same effect in rear. So looking at this rearing barn, this is also on our site. This is, you can see a ramp here, you can see a ramp here, and then this is a schematic. So now at 3 weeks of age, the birds are going to be contained, largely they're going to be in these lower areas. Okay, so you're not really going to see much movement, but even at 14 weeks of age, now that the birds are, well, they can easily jump from here to here, they still, over 80% of their transitions are made with the ramps. So if we look at this, okay, the birds prefer the ramps. When you look at their biomechanical abilities, they're not a hummingbird, they're not a sparrow, so they probably are not able to use the ramps, and it results in benefits, say, reduced keelbone fractures. In this new paper that I mentioned, we also see benefits in terms of feather quality, foot health. So not only are ramps good for keelbone fractures, it also seems to be good because that's what the bird wants. And this is what I mean about building the house around the bird. So, I guess because we are taking a non-traditional view, if anybody has any questions about that, please let me know. But I would say I'll end that section. The idea is that we should be building housing around the birds by knowing not only how it affects obvious things like do the birds have fractures, but also their abilities, as well as their preferences. So I next wanted to talk about something which I've focused on a lot, this idea of individualized behavior. And now I wanted to start off and say, well, this is a long established theme by many people other than me. So I want to give them credit. You know, this idea of, say, coping strategies and even incorporating the individual level observations into your statistical designs. The idea of animals having an individual effect upon other animals. So we've long known that individuals are important, and that's where we should include our observations on. No doubt about that. That's not what I'm saying. Or I'm not saying anything new here. But what I do think is the modern day challenges and opportunities mean that we need to think about individualized behavior very differently. So for one, massive group sizes. Maybe 50 years ago when we had animals in cages, it was easy to do individual observations. But with Maxine's presentation, where you've got 40,000 birds, those are 40,000 birds, but you also need to consider that's 40,000 individual birds. Okay, why are some birds piling? Why are some birds not? So we need to consider that with the increased size of poultry flocks, we also need to find ways to consider them individually as well. And it's more difficult to do that. There's also this idea of individual profiles. And here, I'm going to use this term omics, which I don't know how many people are familiar with, but whether it's genomics, metabolomics, there's this idea that we have this individual level information and we can use that information towards improving the animal. Now, I think what is not here is you often hear about metabolomics or genomics, but from a behavior standpoint, we also are collecting all this information about the individual level data. And I can show you that, how we're doing that in the next few slides. So specifically, with all this technology that we have and computing capacity, I can place sensors on animals that can tell us exactly where it is, its body temperature, what behavior it's performing. So from a behavior standpoint, we can also generate comparable levels of data compared to, say, genomics or transcriptomics or all these other things. So in behavior, we're also able to generate these massive data sets. So what I think is this is really a massive problem because we're having these issues like piling and keel bone fractures, but it's happening in these massive flocks, which are so difficult to observe. Some animals are piling, some animals are not. How do we handle that? And I would say actually my talk is obviously on poultry. I think this approach really applies to all livestock species. So if there's any pig people or dairy people in the audience online, hopefully you guys should tune in as well. But I think some of the ways that we look at our data also applies here. So I'm going to focus on individual observations. Initially, I was really focused on external use, how the birds use the external environment. And I think this is where I first came across Peter and her colleague Hannah. So this is a graph from a colleague of mine, Sabina Gebhardt. And then when I was at the time, actually, I was also in the UK, we had similar data as well. But what we saw here is that this is a percentage of birds or the percentage of individual birds that are using the outside area, specifically the winter garden or the free range area. And what we could see here is that there seemed to be this binomial distribution where some birds were using it and some birds were not. And I found that really interesting. And it got a lot more complicated and more interesting after that. So again, to go back to this graph, where we've got these 5 zones, so the highest tier, the middle tier, the lowest tier, the floor area, and the winter garden. And now, with our technology that we've been using, we can actually track an animal's location in those 5 zones. So over a 24-hour period, I can know where that animal is. And that's shown in this graph here. And this was published in, I call it the haystack paper. But what you have here is this is one particular animal on day 1, day 2, day 3, day 4. And on the y-axis for here, you've got those 5 zones. And on the x-axis, you've got the time of day. So this is hen number 1 on day 1, day 2, day 3. Now what I was really interested in is these highly structured profile of this animal. Every day this animal seems to have a very similar pattern. So you can see here it's at the lights come on, it's up at the highest tier, and then it comes down, and then around here, probably about 2 hours later, it goes to the middle tier, probably to lay an egg. And then you can see this similar profile. So this animal seems highly consistent. So I don't know about you, but I normally keep a pretty good routine. You probably do as well, and so do the chickens. However, we need special technology to actually see this. Now, when we take those same technology and we examine different birds, but in the same pen, different day, we can see very different profiles. So this here is Peta. Every morning she gets up pretty early. She lays an egg pretty regularly. At 10 o'clock, the pop hole opens and she goes outside. Turns out that Professor Hemsworth over here, he's getting a bit older. He's perhaps he is less active, right? But he's consistently less active. Whereas Helen Hemsworth, his daughter, is very active. So she is going up and down, up and down, up and down throughout the day. So these are really interesting profiles that we could see. And I think what struck us is that not only are these individuals consistent, but they're also very different from each other. And so I gave this term, well, I say behavioral structure, and these highly individualized profiles. So moving forward, when I showed, I've never seen my boss excited until I actually showed him these graphs. But you need to move on from there. And you can't just showing, oh, these are really cool graphs, they look really different. So we've, I guess over the next 5, 6, or last 5, 6, 7 years, we've been working on, say, how do we differentiate? How do we show that this animal, how do we show that Paul is different from Peta, is different from Helen? So I'm going to give her just a quick overview. We've looked at, say, daily composites, as well as some variables with specific times and locations, and then I want to talk about how that applies to welfare. So this is a paper that was published by a recent PhD student, and so what this is 4 hens, and she looked at the movement, say, over 8 weeks, and this is a representative graph. Say this is week 2, week 3, week 4, and this is the first weeks that the animals are actually in the barn. And what we could see actually from early on is there seemed to be about 20% of the birds that had very little movement in this initial period, and we wanted to quantify that. I think so much of this is, when we saw this, we could see things, and it just, as you peel back the onions, the layers, you just find more things, which has been really exciting. But how do you differentiate these things? So many things that we've seen, it's like, oh, okay, we didn't expect to see that, let's build an experiment around that. So here, we actually saw this where these different animals, hen 9 is moving from the very beginning, hen 145 doesn't move at all. It stays in the top tier the whole time. We initially thought, oh crap, our equipment isn't working, but no, it was working. It's the bird. So what we did, or what she did, is created a movement component, a single variable for each day. And it's composed of the duration in each zone, so there's 5 zones, the transitions into each of those 5 zones, the vertical distance per hour, so how many times she's transitioning per hour, whether she uses the Wintergartens in the first 15 minutes, and where she sleeps. So she combines all of these things into a single variable, a component for one day. I think this is interesting. We call this a movement variable. If animals move a lot, this variable is going to be high. But you could also include other principal components which we're working on. But for this, as an example, this is a movement component. And then what she did here is actually, so this is the time, so the first 55 days in the barn, and on the y-axis you've got the principal component value. And what you got is these 4 hens that I've showed here. And this is a really cool thing, is that you can actually see the differences here. So looking at the principal component, you could see hen 9 here, versus in the yellow, versus the green hen 145. So you can see they're very different in terms of their average values. And that's shown by this graph here, where you've got the principal component on the x-axis. So you can see here, hen 145 is generally lower than hen 9 here. But we also started to look at things about the predictability. So it turns out that Paul got up very early this morning, but the next morning he got up really late. The morning after that, he gets up early again, and he's very variable. Whereas Peta, every single day, she gets up at 4 o'clock, has breakfast, and is in her car driving to work by 6. She's very regular. We can also see that in the animals. So looking at the blue line here, you can see this animal, day after day, it's very variable. Whereas, actually, the green animal is pretty consistent. So these are various different ways we can look at this data. Now, I think this is obviously interesting for poultry, but why poultry? It could be in any species. It could be PETA, it could be humans. It could be pig data in terms of when they access a feeder, whatever, cows when their milking percentage. So this is looking at variation over time. So we can see this variation, we can see there's lots of variation that exists. And I'm going to actually then come to actually how we can then relate that to various things of interest. But just for now, hold this thought that lots of variation is there. And we now have techniques to actually take these really cool graphs that look really different, and we can actually show how they are different and quantify that. The other thing that we've been able to look at and is actually specific events that are happening and how animals respond to that. And with this, see, we just have these 5 zones, and this is what I mean by small resolution. We just have 5 zones, and people have told me, well, you could use GPS and track exactly where the animal is, but within a commercial barn like what we have, the signal gets distorted. There's lots of technical problems that I can't explain to you because I'm not the technician. But just understand that it is, of all the various tracking systems we use, they all have problems. So we can't really tell what the animal is doing or how much in proximity. All we have is that the animal was in this location at this particular time, but that we're very confident about. But with that information, we still can extract really cool results. So this is the data broken up into those, we have 5 zones, and I'm just showing you 4. So the top tier, the nest box tier, the lower tier, and the littered floor. And this is a percentage of animals that are in that location at this time. This is broken up into 1-minute intervals. So, and then what these green lines represent, so that's these specific areas. So one thing is laying. And this is percentage of animals, so you'd expect that actually as the animals, within say 2 hours of lights coming on, they access the nest box. And that's what this is showing, is this is the percentage of animals as the day progresses. It peaks at about 40% of the animals are accessing that tier. Now, we don't know what they're doing in that tier. They could just be going up there and just peeking in. I'm not gonna lay an egg here, but we think they're probably laying an egg. The other thing is that looking at feeding. So we don't know what the animals are doing, but we do know that there's only feeders at the top tier and the lower tier. Now, these green bars represent when the feeding engine comes on. They always have feed. There's always feed in the feeder. But we do know that the birds like fresh feed, the bigger particles, and that's pretty uniformly seen. So now what's interesting is when the feeder comes on, as shown by these green lines, you can see a dramatic increase in the number of birds that are accessing the lower tier where the feeder is. So there's a big drop, a big increase here, 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 and here, right around that green bar. Similarly, you can actually see there's a drop in the floor area. The feed comes on, the birds hear it, and they all run up to the lower tier. So these are kind of timing events that we can see that are happening to the birds. Now, not all the birds can access the nest box at the same time, only about 20%, and probably the same percentage for the feeder. Only about 20% of the birds can feed at the same time. So it's interesting for us that we can now see which birds are feeding, how do they organize themselves. Simply, Peter actually tells me that she comes really early to work because she wants to miss traffic. So she is coordinating her schedule about everybody else's. And it's interesting to think whether the birds are doing the same thing and what's driving that. So this is the kind of thing that I'm hoping to investigate over the next 10 years or sooner. But the idea here is that we can pick up these types. Uh-oh, I just clicked a button and now it's, shit. I just turned on the volume, I think.
Oh.
There, yeah, that goes, yeah. Okay, don't click anything. So yeah, I guess my point is that there's lots of information here. There's very little we actually know about what's happening, but because we're recording every single animal of 24 hours a day, there's still really interesting patterns we can pick up. So, what happened? There we go. There we go. And then this is kind of a blow up of this screen in case you couldn't see it very well here. I've blown it up, and this is the animals that are accessing litter, and this is animals that are accessing the lower tier. So it's really an interesting pattern. You can see this spread out, and you can actually see there's probably a wave of animals that are first accessing the feeder, and then a second wave of animals are coming through. So this is some really interesting results. The same thing for the pothole. Not all animals can access them with regard to the same time. They have to negotiate that. How do they? Do they look and say, well, I never want to hang out with Peter, but I really like Helen, so I'm going to hang out with Helen. Like, how are they figuring this out? And it seems like they are. And I think it's really interesting for us to figure out how that's happening. So that's really what we have seen. But now, what have we actually learned? Because it's really interesting to see these really interesting graphs and things, and it helps us inform the fundamentals of the animals. But how does it actually relate to their biology, their health? And these are some snapshot of several pictures that we've seen. So this is firstly movement profiles in health. And this is keelbone fracture severity on the y-axis. And on the x-axis, this is predictability. So this is that movement that I showed you. Let me go back here. This graph here. The predictability is how much variation you see from day to day. So blue would have very high predictability. No, sorry, it's confusing. Blue, which is all over the place, has very low predictability. It's difficult for me to predict where that animal is going to be the next day, whereas green is very predictable. So which is better for the animal? Well, it turns out in terms of keelbone fractures, an animal that is more predictable is going to be more likely to have keelbone fractures. Now, what's really interesting about this is predictability in the first 5 weeks in the laying barn, but this is measuring keelbone fracture severity 40 weeks later. So now, whether that's mechanistically related, we don't know. But basically, if we want animals to have less keelbone fractures, we want them to be less predictable, a more varied schedule. So Peta, who comes into work every single day at the same time, she's more likely to have a keel bone fracture. So we actually want Peta to come at all different times. Now, this is just about keel bone fractures, and who knows whether it's mechanistically related, but this is a really interesting relationship. And this is only with 80 birds. New studies we have coming up are going to be with like 1,000 birds or even more. So we can actually hopefully strengthen this prediction. So that's keel bone fractures and the predictability. None is toe injuries. So, and we have, this is a problem in Switzerland where we see the birds have injuries and we don't know if they're pecking themselves or other birds are coming. But we've developed a scale. We're going from 0 to 10, and that's not going to show you very well, but this is a toe injury here, okay? And I think just like piling, you have good producers, and then for some reason, anywhere from 50% of the birds in really healthy flocks, good producers, have these toe injuries. And the bird, because they keep picking at it, will bleed out and often die. So this is definitely a big problem we see in Switzerland. So what we were interested in is when the birds have these toe injuries, how does it affect their movement? And that's here. This is looking at just daily transitions. And you can have 3 colors here. Birds, the blue, no toe injuries, gray with minor, and then red, which is severe. And what we were interested in is we could see here, there was and this is a missing data here. This is because we were doing health assessments, so we're not tracking the animals. So we were actually quite curious because here we see big differences, but what comes first? Is it because maybe the animals with severe toe injuries are moving very little? And we actually go back in the data, back and back and back, all the way to 150 weeks. So basically about 100 days before, and you actually do see them coming together. So they're one population here. This is just preliminary, but I think there does seem to be where the birds start to fall off here, and then they drop off really severely. This is probably that as the birds have these toe injuries, we see them sequester themselves in a particular area of the pen, hiding themselves. They pick up their toes so other birds don't peck at them. So we're seeing different behavioral effects. We can also see the toe injury affecting production. Now, we can't actually measure egg production, but remember when I mentioned earlier that we can actually see, well, does the animal go to the nest box area? And we've actually been able to show, we would expect for egg laying, the animal needs to go to the nest box area, and within the first 7 hours of lights coming on, and spend there a 30-minute period. And we've been able to connect that behavioral data with production data from our genetics colleagues. So we think this behavioral proxy is a really good indicator of egg laying. And what we can see here is this is the amount of eggs laid within a certain period on the y-axis. And this is on the x-axis, this is different pens, but these are basically birds with, I'm missing the label here, sorry. These birds have severe toe injuries and these birds not. So the birds that actually have the toe injuries are producing less eggs. So again, I think this is a really good example. We can't measure egg production, but we have a behavioral proxy that can inform us about this. These movement patterns also have a biological basis, so we can definitely see differences in behavior. This is a recent paper where we have these very special genetics that we can actually quantify the heritabilities of these behaviors. So you can actually see this in this table. This is the amount of time that the animals spend in each of these zones and looking at the heritabilities. And what this is, if you think about all the variation you see, some is due to genetics and some is due to the environment. That's an oversimplification, but you can understand it. And this suggests, so if you want to breed birds that spend a lot of time in zone 7, this is a relatively high heritability, 27%. So 27% of the variation is actually owed to genetics. So what we're showing here is that because the durations basically dictate all these more complicated metrics, we can actually breed for these behaviors. So when I provide this information to the genetics companies, they can actually now breed for birds that spend more time in the floor or more time in the outdoor areas. We can also see that the birds move in groups. So if there's, in these pens, which are, say, about 225 animals, they're all moving around, and they all look the same, but what's actually driving their movement? So if we look at actually when the animals appear in the same area together, That's what this shows. And are there any connections? They're all basically all connected. So this is the 225 birds in the pen and their connections. But if we focus on the bonds that are particularly strong, we can see some elements actually being stronger. So this animal seems to be more connected to that animal. And then if we look at, say, a hierarchy, so within that population of 225 birds, about 10% seem to have particularly strong connections over the entire 36-week period. And this looks like somewhat you would see of the traditional social networks, where there's some animals that are more central and more connected to others, and say these animals are on the fringe. Now, I'm really interested in this because you have these techniques of this concept of, say, social buffering. If you have lots of friends that you can relate to, you tend to not get sick. This is from human data. And we would expect the same thing with the birds. Are they actually able to form connections and social connections with the other animals? Well, we have evidence that they're moving together. We next want to ask, well, are these connected in some type of social manner? This particular bird here, or these birds in this group, are they more likely to not get sick or to recover than, say, birds that are not connected? These are the kind of questions we hope to answer in the next 10 years. Or 5 years. Also questions that I put this in specifically, I think this is important for Australia. Where do the birds actually want to go? So in Australia, you tend to just have, from what I understand, a free range setting. In Switzerland, we also have this idea of the winter garden, where the birds can actually access, it can be outside, but it's still covered. They can benefit from the sunshine, the fresh air, but it's covered, so they're protected from migratory fowl, predators. If it's snowing or raining, they can still go outside and not get cold, not get wet. And so this is a study we released here, where we looked at this is a flock of 360 birds, where they could access a free range, a stone yard, as well as a winter garden, and then inside. So actually, I went backwards. The animal would be inside, it could access the winter garden, then go to the stone yard, and then go to free range. And this is basically over a 40-week period, the proportion of time that the animal spent in those areas. So most of the time is spent inside, and then you see a lot of time in the winter garden, and then a little bit in the stone yard. What I thought was really interesting is that a lot of animals do access the free range, but the vast majority of time is really in the winter garden in terms of external areas. So I think this is interesting in terms of if we want to provide animals to access to outside areas, they seem to like the winter garden. So just like the ramps, how should we provide these environments? The winter garden can be expensive, but in terms of providing the animal protection during avian influenza outbreaks, the animals can still access the winter garden. The free range is really exposed. You're exposed to the rain, the predators. Yes, the animals will use it, but they definitely prefer the winter garden. And this is the last thing I'm going to show, and I probably should skip over it because I'm going over time. So just wanted to catch up that I think there's incredible tools to assess animal individuality. There's the genomics and other omics that I think are really fascinating, but we can also generate these incredibly large data sets about behavior. And I think this is going to provide us a better understanding of animal needs towards building these welfare-friendly environments. So the last thing I wanted to talk to, and Peter, give me the yank when I'm going too long, is about this hen track project, which is taking these tools that we've been developing with technology and about phenotyping and examining behavior and applying it towards breeding. So as many of you know, and so Maxine talked about piling, cage-free systems have welfare problems, and the public probably doesn't really like this. They're supposed to be welfare-friendly. We moved from cage to cage-free because we think they're better, but we have lots of these problems. So whether it's piling, toe pecking, or keel bone fractures, and many others, hygiene, other things. There's this mismatch between what we would like cage-free to provide and what the actual reality, which definitely keeps people like Peter and I and Maxine employed, but we also really want to solve these problems. And I think one of the major reasons for this is the way we breed animals. So our current methods are based on using small groups of, or individuals. So you see a trait that you're trying to breed for, whether it's good, lots of eggs or feather quality, but these are all traits that you can easily observe in small cages. And that's why birds in cages do so well. You know, over the last 30 years, we no longer have problems of aggression in cages. We have incredibly high egg outputs. The birds are very feed efficient, all the good things about that. But then when you take the genetics that perform well here, and then you place them in a big aviary, this is where you get all these problems. So this is certain things that do well here don't translate well to here. So let's say feather quality. Birds that have good feather quality here may not have the same feather quality here. You think about bone health. The bird which is here and then needs to get up here to roost is going to need very different bones and muscles than what is here. And outdoor use, if you're looking at birds here, how do you know if this bird is going to go outside? So these are traits that you need to quantify and so that you can ideally build a bird that's better in this environment. So the goal of the Hen Track project was to provide methodologies for the phenotyping and selection of laying hens that perform robustly within cage-free housing systems. Yeah. It's basically trying to give the breeding companies tools so they can select these traits in cage-free housing. So we have these really special hens where generally, for those that don't know breeding, at the top of the line you have these pure lines, and then they breed the grandparents, the parent stock, and then finally the commercial laying hens. So one pure line bird can actually lead to something like 400 million eggs. So there's this incredible hierarchical structure. What we have as part of this Hen Track project is they've given us basically the daughters of the pure lines. So we can then measure things here and then feed the information back to the company so they can affect the breeding. So now they'll typically, the genetics companies will be looking at, say, feed efficiency and feather quality, and they're measuring those things, but they're doing that in cages or small groups. We're using these cage-free systems and using the tracking technology that I mentioned. So we can measure things also like feather quality, but also things like winter garden use, as well as the basic health of the animals. So we're working with two of the world's largest genetics companies that's expected to, well, they control something like 90% of the laying hen genetics outside of China. So it's this massive effect. So it's a really cool project. I'm going to skip over this and just say that they're giving us these, say, various hens that come from various sires. So we can look to see when these traits, how is it influenced, say, by family, across generations, because we have multiple flocks. Sorry. So I want to end on this piling information. So like Maxine showed, we did similar work in Switzerland where we were looking at, we know that piles occur, and then they seem to occur more often in white hens and at what time of the day. But we really wanted to know about the individuals. So we can see in the group piles here, but we want to know, is this individual always piling? So we did some preliminary work that Maxine came over and worked with us, and we were using these antennas underneath that can locate these particular individuals. So looking at this graph, this is on 3 separate days, and these are separate piling events. And on the x-axis, you have each animal. And this is the number of piles of that, or the number of times that animal is involved in that particular pile. So what this is showing here is on the, sorry. So we basically started with each pile, whether the animal was present at the start, whether present at the end, and then whether it was present at 25%, 50%, or 75% of the way through. So that's 5 time points. And then the colors coincide with how often the animal was detected at that pile. So for piling event 14, this animal here was seen at 4 of those time points, whereas this animal here was never seen. And this is for pile 14, 13, and 12 on the 26th of May. This is for the 16th of July and then the 25th of August. So what we can see here is that there are certain animals that seem to be piling very often, whereas some animals are never piling. So, and that led us to Henpile 2.0 with this hen track project, where now we're doing this, thanks to Australian eggs, we're quantifying this within our flocks, the hen track flock. So we're trying to ask basic questions like who is always or never involved in a pile? Who is always there at the start of the pile? Who perhaps in the middle of the pile leaves? And where are the birds before they join the pile? We're also looking at, say, the bird's response to a novel object as well as a novel person. And we're also considering automatic detection. So for this types of things, because it's involved in the overall hen track project, we can calculate the heritabilities. What's the genetic controls for if a bird always is involved in a pile? So I wanted to say that we're doing some really cool technology, but it's not a silver bullet. Technology companies will always tell you, yeah, it'll solve everything. Don't believe that. So, and the last thing I just wanted to show, if I can, is here. So what this is our data. Sorry.
If you go here, and then pick your, this one, yeah.
So what this is, you can see the birds here, and then what we have underneath is that now as the birds are moving, these digits that flash up, this is we've taken our tracking data and synchronized it with our video data. So as the birds are moving, you can actually see that pop up. So we have a pile here, and in theory, we can determine which birds are involved in the pile. So this is some cool data that we have. And with that, I am now done. But that's not my slide, and I don't know if you can see that. Back to here. So I just want to say that I'm really eager. If anybody has any suggestions or would like to collaborate, it's really important that our interventions are based in the fundamentals of what's causing the problem. I think individual behavior offers a really exciting frontier for animal welfare. I think the sensor technology and technology in general is really interesting, lots of opportunities, but we still need to understand what drives behavior. So we need to take the technology people and combine them with the people that know behavior, the producers as well, obviously. So I just want to say lots of thanks to lots of people like Peter Taylor and Maxine Fogarty, which obviously are some great collaborators and lots of funders. And I can turn it over back to Peter there.
Stay here. I think it's one thing working with Mike, it's always hard to get him to stop talking, but I think that's because you've got so many wonderful, interesting things to say. I think we have a couple of short minutes. What I want to do is invite a couple of questions, and Mia's going to do that for Mike, and I think we can continue the conversation offline as well. So
firstly, there's been a couple of comments that toe pecking has not been seen here in systems here so much, including from someone who's going to catch up with you at PICS on Monday and Tuesday and chat to hear more about your experiences. But Hannah asked, does variation within individuals occur more in housing systems with more behavioural opportunities and less restriction? So say consider a behaviour like feeding or nesting, which would be available also in a more restricted housing environment.
So I think that's a good question, and part of the problem is that we have a great housing system, but it's just one. So it's really difficult for us to say these individual levels across other housing systems. We're still just exploring. Like you look at the graphs and you can see they're really different, but we don't really know what's driving it. So going ahead, we do plan to say diversify the enrichment. So provide more pecking stones, more opportunities for feeding, even insects. And we would like to see how it drives that variation. But at this point, it's so exploratory. So I guess there's not really a good answer for that.
And then another question, I guess, is how similar, as from a behavioural and motivational point of view, are toe pecking and feather pecking as behaviours?
Good question. So I think this is big. For feather pecking, we know that other birds are initiating it. So Helen pecks on Peta, and we can see that. But the problem with toe pecking is we don't know how it starts. So it could be that Peta has dry skin and she starts pecking herself, which then attracts Helen over, and she sees, what is that? And Peter has caused a little bit of blood, which then attracts Helen, and she starts pecking at it. So we don't really know how those things happen. So I would say they could be or originate from similar places, but they also can be very different as well.
Great, that takes care of these questions. So free to the room.
Thank you so much, Mike. I will just say for those who will be at PICS next week, do try and catch up with Mike, reach out. He's really great at doing Chris Isaac at karaoke. So opportunities to do karaoke and talk about chooks. So please do reach out and catch up with Mike. Thank you so much. Yeah. I have underestimated, as always, just how much we have to get through and how much exciting data there is out there. So I'm going to just fly with this for a moment, and instead of getting us back to doing some questions online, we still want to hear from you. And I know it's difficult when you leave a seminar to remember to give us some more data, but we will send out the recording, a link to a recording. We'd love to hear, some people are putting in their responses here now. What stakeholder group do you belong to? And you're going to get mad at me because I'm going to fly through these. We want to hear, should I leave this a minute? What have we got? We got 2 minutes. We can do this, seminar team. I'm going to move through to the next. I want to hear about your level of concern about piling and smothering. This, I think, I'm going to turn into a Qualtrics and send out with the recording link. What are the things that particularly have you concerned, maybe on your farm, in your industry, or maybe even just in terms of what we've heard today? We want to hear about what poultry welfare research you want to hear investigated. Maybe one I'll leave with today, because the seminar series are back. These will happen quarterly. And we'd love to hear what topics you'd like to hear about in the future for animal welfare science seminars. Mia, straight in there with the dogs. So with that, I really want to thank both Maxine and Mike for sharing such incredible information today, for their time, for coming all the way out. For Mia, for her incredible behind the scenes and chat work, T for AV, all of those involved, and of course our participants online. Thank you for being here, and please keep an eye out for some important seminars coming up. As Lauren mentioned, the next one we aim to pay tribute to Andrew Fisher's contribution to animal welfare science, and so we will see you online and hopefully in person. Thank you.